Every thought, memory, and feeling flows through a neural network. To decode the mind, we must map how billions of neurons connect. This requires imaging hundreds of billions of synapses in high definition, a task requiring dizzying datasets and obscenely expensive equipment. But now, researchers are using AI to select where to scan, which they call smart electron microscopy, to one day reach a moonshot goal of mapping a human brain.
To understand how brains work, we actually need to understand what neurons and synapses look like. An electron microscope allows that kind of resolution, but you need a multi-beam microscope that most labs can't afford. And so the question is, how do you take a regular electron microscope in itself on the order of $1 million and turn it into the equivalent of a $10 million parallel multi-beam microscope? How can you do that? And the solution uses machine learning to actually speed up this kind of computation.
I'm a neurobiologist interested in the structure of the brain, and I confront a problem of the big and the small. The big being that a brain is a reasonably large piece of tissue, and the small being that in order to make sense of it, we have to see the finest details, which require very high resolution. There are a few machines that can go very fast, but they are insanely expensive, and if you had to do a whole brain this way, it would be an enormous expense.
Now, pixel in fMRI is about a cubic millimeter. And in that cubic millimeter, there are about 100,000 neurons and 1 billion synapses. Electron microscopy allows you to look into that cubic millimeter.
If one images it entirety, cubic millimeter of brain, the size of the head of a pin, it's about 1,000 terabytes or a petabyte of data. It takes a long time to get that kind of data. And so we are always seeking ways of doing that as quickly as possible.
The goal of this research is actually to get a full connectome map of a mouse brain. And we could use it across the world in many labs. But to get the mouse connectome, one lab cannot do this job. If you don't manage to speed this up, this project is just going to take too many years. We need to get tens or even hundreds of labs to collaborate to do this. And they don't have the ability to buy expensive microscopes. We want labs that have a single beam microscope to be able to participate as if they had a very expensive parallel microscope so that we can actually together get a mouse brain.
So in connectomics, our goal is to be able to segment the tissue with high fidelity. The main issue is the time of the imaging and the computation that it would take to do the reconstruction afterwards. The idea of SmartEM is to try to accelerate this, so this could be done by first training a machine learning algorithm to detect errors from fast images. Once the machine thinks that by going too fast it may have errors, it would deflect the beam to these regions that may cause the error and re-image them slowly with high resolution until the error or the ambiguity is gone.
Researchers at MIT and Harvard are using SmartEM to map the neural tissue of a jellyfish in under a year.
In my lab, we use the jellyfish, Clidia hemispherica, to study aspects of how the nervous system controls behavior. One key piece of information is how all of these neurons are connected to each other. So that's the connectome. The way that we are doing this is by taking this jellyfish and cutting it into 20,000 very thin sections. Using classic EM approaches, that would have taken about 9 years to image. But now, because of SmartEM, it's only going to take about 6 months. The particular challenge with the jellyfish is that it's a huge volume, but most of that volume is actually jelly. So what SmartEM is allowing us to do is just avoid imaging all that jelly at high resolution and just find the places where the neurons are and get the information we need from that.
So what we see over here is jellyfish embedded in a resin. If we zoom in, we will see that this is actually a multi-resolution image, which has details only where the biology matters. Over here, I just defined from this ensemble 6 rectangles. The microscope will be instructed to find these parts, deflect the beam, and take them at high resolutions. And the scope will be instructed to take the job. Over here, you can also see the transition between the areas that are not imported with the more imported ones. You can see over here a few mitochondria, and the one over here. And this is saving a lot of imaging for them because you see that the image is very sparse. You don't really need to image in the pixel entire resolution.
For us, the holy grail is getting the entire brain of a mammal. And that, we're getting to 1 million terabytes, for example. That kind of data requires going way faster than we can currently go. So one is hoping that tools like the SmartEM, which is relatively inexpensive because so much of the effort is in the artificial intelligence that allows it to scan quickly, will allow us to have enough machines working in parallel that one might be able to do a whole mouse brain. And at some point, maybe a whole primate brain, and looking into the deep future, a whole human brain. The kinds of knowledge you need to build this kind of microscope requires understanding of artificial intelligence, coding, and microscope technologies that are themselves quite recent. And so this is an interesting collaboration between older and younger scientists. I think the younger scientists might say it could have happened without us, but it couldn't have happened without them. Maybe the way I would say it is that young scientists absolutely had to be involved in this because this is very cutting edge.